Good afternoon. I'm Dr. Anita Pepper, Vice President of Institutional Advancement at the Wistar Institute. Welcome to the Wistar Institute's Town Hall. The goal of today's event is to give you all a sneak peek as to what's been happening at Wistar during the devastating COVID-19 global health crisis. We're proud at Wistar that we've been able to stay true to our mission of sustaining our cancer research and educational platforms during this pandemic, while quickly advancing our research on vaccines and therapeutics, trying to curb the COVID-19 pandemic. I'm very pleased today to be joined by Dr. Dario Altieri, Wistar's President and CEO, and Robert M. Pennyfox, Distinguished Professor and Director of our NCI Designated Cancer Center. So good afternoon, Dario. How are you?
I'm good, Anita. Hope everybody's safe and healthy, and I'd like to extend my warmest welcome to everybody that is in attendance today. And I hope that everybody there is also safe and healthy in these very challenging times.
It is indeed, and thank you. So I wanted to start the day kind of on the macro level and go into the micro level as we move in. So as scientists, we've never seen kind of this public display of scientists' work and how the actual process works as we have during the COVID-19 pandemic. Day to day we get to see an update on where a vaccine, a therapeutic, immune system, people know the words antibodies now. So can you tell us how all of this is changing what the public understands about what Wistar does in scientific research?
Well, it's been certainly like being under a microscope for the past 6-7 months. This is not typically what scientists are used to. Sometimes what has been said about COVID-19 and SARS-CoV-2 has been utilized for political reasons, has been weaponized, has been politicized, even beyond the intent, really. We as scientists ask questions, we try to solve problems, to find a solution to complex events, and I think SARS-CoV-2 is almost a perfect example. This was a new virus. It was characterized by zoonotic transmission. That means that the virus jumped from one species to a human. And it was a completely new virus from the epidemiology and the molecular biology. And we have never been really exposed to anything like it. And it took us some time to learn. And as scientists, we formulate hypotheses. We try to design experiments in order to disprove that hypothesis. And if at the end of the day, we look at the data and we formulate potential conclusions. In this particular case, I would like to say that we're still learning. We're still learning about the demographics of the people who are most affected by the virus. We're still perplexed by some of the ways in which the virus spreads. We do know that there are these super spreading events, and we also know that comparable locations in other places of the world with comparable geographics and climate are in fact not exposed, don't show the same level of exposure. So it's been a process, and it takes some time. So nobody has a magic wand, and the scientific process is one that is based on data collection, formulation of hypotheses, and hypothesis testing. And I hope that that's really the message that we've been able to convey over these past few months, that we help finding solutions, and this is really a scientific solution.
Absolutely. And I think most people don't understand how frustrating the pursuit of science can be, because there are twists and turns, and normally that's not on display. Normally we get to a place where it's an endpoint of look what we solved, but now you're actually seeing the whole thing on display. So
we do. I'd like to say, though, and you probably agree with this, Anita, I would like to say that ironically we learn more from an experiment that doesn't go the way we planned than from an experiment that goes exactly as anticipated. And we learn more from our mistakes and from our errors, and particularly when we're confronted with something entirely new like SARS-CoV-2.
Absolutely. So when thinking about Wistar and our mission of basic research, how does this change how Wistar does its research? And kind of part two of that is, how do we actually, as a basic research institute that's completely independent, we're not connected to a hospital, a university. We have lots of partners, but we're totally independent. So how does this change how Wistar does its science and how we are trying to advance human health?
Well, ironically, it hasn't really. I mean, these are our values. What you just articulated are the values that have permeated the Institute since its inception. We are a single mission organization. We're all devoted to the understanding of human diseases. And we prize the scientific methods as the way in which we have been able in the past, and hopefully in the future as well, to solve very complex problems. SARS-CoV-2 is an example, but obviously, complex diseases like cancer, which is another main topic of the Institute, has been our target for the past 127 years. And so, in essence, we have repositioned efforts in order to be at the forefront of new discoveries related to SARS-CoV-2. But in essence, the approach that we have taken, the strategies that we have utilized are the same that have directed the Institute in its quest for new knowledge about human diseases.
Yeah. So I think it'd be helpful maybe for the audience to share a little bit about Wistar's framework. So we are unique, we are a small engine, and we kind of have 3 pillars. So maybe if you could tell a little bit about that before we dive into what they actually are, that would be helpful.
So, it's an interesting model because research institutes like Wistar are not universities, are not colleges. We don't give degrees, we don't admit students to our facilities. As you pointed out, we collaborate with health systems and hospitals, but we are not part of a health system or a hospital ourselves. So ironically, to use the jargon that I was using before, as a single mission institution, we only do one thing. And that's, in essence, that's the appeal, if you will, of having a dedicated entity that is solely focused on biomedical research that involves the study of human diseases, pathogens, cancer. And Wistar history has been and continues to be very focused on translating those advances that we do at the research bench into cures and vaccines and therapeutics that are safe and can make a difference in people's lives. So that is really a departure, if you will, from a more traditional university settings, where there is not just one mission, there are many missions that the organization has to accomplish every year. For us, it's research, and like I always like to joke, at Wistar even the bricks want to do research. So we're very proud of keep doing that.
All science or nothing. So you mentioned vaccines and the Vaccine Immunotherapy Center. So as we're diving in a little bit, let's start there. I know there's a great deal of interest and attention right now on what's going on in our Vaccine Immunotherapy Center. And this is clearly Wistar's moment to shine and to rise. So pandemic preparedness and response has been part of our mission pretty much since we opened in 1892, 1918 flu. 1960s, we created the rubella vaccine that halted the epidemic then. So I wanted to talk about how Wistar has tackled fighting COVID-19 and maybe how we've shifted our focus in the Vaccine Immunotherapy Center and pivoted to really focused on COVID-19 preventatives and therapeutics. So perhaps you could tell us a little bit about what we're working on, some of the new technologies. This is a very large question. Some of the new technologies like nanoparticles, and an update where the research is focused right now. And we'll give you a slide in the background to try and help do that. Yeah, there we go.
And I think the slide itself conveys the message of what has been the spirit of the Institute when we started tackling the COVID-19 emergency and the pandemic. Our first order of business was obviously to keep everybody healthy and everybody safe at Wistar, our scientists and everybody else working here. And as the slide indicates, we have taken a multidisciplinary approach. We've been trying to tackle the same problem from multiple angles. Certainly The vaccine is one, which will continue to be very important now, especially that some of the largest phase 3 efficacy trials are approaching the moment in which interim results will be reported. So there are two Wistar vaccine candidates that have been developed, utilizing two completely different strategies. We put tremendous effort also on therapeutics, trying to develop a platform for inhibition of, or at least decrease the entity, the extent of COVID-19, of SARS-CoV-2 infection, utilizing small molecule approaches. And the nanoparticle technology that you mentioned a moment ago, Anita, is one of our newest approaches, one of our newest platforms to try not simply to package a potential vaccine for delivery into a more easily manageable delivery mechanism, but actually to engineer particles so that they would be presenting antigens, viral-related antigens, that would make a much more significant, much stronger immunogen for our immune response to develop antibodies.
So from my understanding of a nanoparticle, you're almost taking a shape and loading it with hundreds of vaccines and then having it go attack, right?
That's exactly right. Right. And that is in essence the appeal of a potential advantage of concentrating a large amount of antigens that come from the virus and our body would recognize as non-self, and therefore mount an immune response to it. And we're working feverishly on that technology to see whether it could provide also an additional angle for delivery of SARS-CoV-2 vaccines.
Interesting. So as we're clearly entering a new wave of increased cases nationally, can you tell us about other aspects of the vaccine and immunotherapy center work beyond the virology and the vaccinology and the nanoparticles and therapeutics? But I'm really interested, and I've been hearing a little bit from epidemiologists about how we really need to focus on bacteria right now. And I was hoping you could tell us a little bit about that, why we need to focus on bacteria when there's a virus, obviously, and really what our approach has been within the Institute.
Well, it's a critical question. It's a very important development of many virally transmitted diseases, particularly respiratory viruses like SARS-CoV-2. And that has to do with the fact that even though the primary pathogen is a virus, and viruses are not obviously sensitive to antibiotic treatment, superimposing bacterial infections could provide truly life-threatening conditions. We know that the COVID-19 is a very erratic behavior. We see individual patients that are admitted to the hospital, they seem to be doing just fine, and yet their oxygen saturation level is dangerously low. A chest X-ray or a CT scan of the chest shows extensive involvement of both lungs with interstitial pneumonia. And so that's already a very difficult and very serious condition on admission. And oftentimes, despite the strictest aseptic conditions in hospitals, that is a very fertile ground for a superimposing bacterial infection, which could have catastrophic effects, obviously, and very poor outcome.
So I'm sorry, I'm going to interrupt for a sec. So when you say a superimposing infection, you mean a bacterial secondary infection to go on top of the virus. So now we're battling multiple things.
That's right. And this is a very common occurrence. It's very common for other respiratory viruses, very common for flu, for instance, where there are still approximately 30,000 fatalities a year just in this country related to flu infections. And so a superimposing bacterial infection that complicates, is one of the complications of respiratory conditions brought about by an initial viral infection, could have very serious consequences. And so one of the approaches that we have been pursuing very aggressively at the Institute is really to try to develop strategies for overcoming antibiotic resistance. This is a big problem. Antibiotics are among the most successful and useful drugs that have ever been made. The problem is that we use them too much, and the bacteria evolve and develop ways to bypass their sensitivity to antibiotic treatment. And obviously that creates an incredibly dangerous situation, because at that point not only you have a superimposing bacterial infection, but you have one that is insensitive to treatment. And so one approach at the Institute and a couple of labs have been working very hard on identifying molecular bases of antibiotic resistance and trying to devise strategies to go around it and restore sensitivity to antibiotic treatment.
So this would actually be new classes of antibiotics to try and treat these secondary infections that we know in hospitals like MRSA have become rampant and almost impossible to treat, or very difficult to treat.
Absolutely, and is a major problem, is an urgent problem, and unfortunately is rendered even more acute by the demographics and the spreading of COVID-19. We know that the so-called nosocomial spreading, virus that spreads in a hospital setting, is very common for this particular condition. And therefore, we need to be able to handle the complications that emerge from it.
Always looking to the future, what's coming after us next, right? It's crazy. So we're a little engine. We don't have that many labs in comparison to most giant research enterprises. So can you tell me, we're advancing 2 independent vaccines right now that are based on different platforms. One is a synthetic DNA platform and one's an adenovirus platform. Can you talk a little bit about why there's so many vaccines being made right now instead of multiple efforts on one vaccine, and what the advantages of having multiple vaccines actually make it to the finish line?
Well, it's a very important question, and right off the bat, I really would like to say that it would be great if we were to forget about sports analogies or the vaccine race, and it's absolutely imperative that we have a portfolio of approaches. First of all, we don't know which one will work. We don't know which one will work the best. And second of all, not every vaccine is created equal. I mean, it's quite possible that one group of individuals at high risk of SARS-CoV-2 and COVID-19 related complications would benefit by vaccination with one platform and not by another. Obviously this relates to the fact that individuals over 65 or 70 years of age with comorbidities and other existing conditions are particularly at risk for SARS-CoV-2 complications, but so are health workers and doctors in emergency rooms and pulmonary units. So obviously these are 2 very different demographics of individuals that are both at high risk of significant viral load upon infection, and they may respond differently to different vaccine approaches. So it's very important that we have options, that we have choices, and ultimately the decision as to which one will be licensed and will become a product will depend on the science and the results of the clinical studies that are currently ongoing. We have seen what happened, particularly related to the SARS-CoV-2 pandemic. We have seen what happens when we try to jump the gun and move towards emergency authorization too quickly. And nobody's fault, of course, even though perhaps was one example where politics started injecting themselves too heavily in a scientific decision. We authorize convalescent plasma and hydroxychloroquine as potential treatment for COVID-19, when in fact clinical trials around the world demonstrated that neither treatment actually benefited patients with COVID-19. So we want to stay away from that. It's going to take time. This is not a situation, a fast food situation where you go to the window and you get your burger. We need to wait until we have all the data and make an informed decision, and it's imperative that we have actually a portfolio of different vaccine platforms to test, to select.
Yeah, no, it's fascinating, especially as you're seeing, as you've said, that the immune system is responding in different populations over such a wide range that you might want a weak vaccine, you might want a strong vaccine, you might not need to boost somebody. So it is, I think as people understand that this isn't going to be a one-size-fits-all, I think it's really important.
I think that, from what we're learning right now, it seems also clear that the protection, at least based on the production of neutralizing antibodies, the protection that is afforded against SARS-CoV-2, against the virus, is transient. It's 3 to 4 months. And so you're looking potentially at a scenario where vaccination against SARS-CoV-2 might have to take place similarly to what we do for the flu every year. You get a flu shot, hopefully. If you don't, please do get it, because it does matter.
Yeah, that is public service announcement. Get your flu shots, everybody. Keep going. So, okay, now I'm going to go to your favorite topic. Not that the VIC and education aren't high up there, but your favorite topic, cancer researcher, as a cancer researcher. So this has been a really hard year for cancer researchers. And so I wanted you to speak a little bit about what's been going on in the cancer research arena nationally, and how has Wistar's cancer research really been affected by the pandemic?
Well, it's been a big problem, obviously. The consequences of the widespread COVID-19 emergency and the pandemic have placed cancer research and cancer care a little bit in a position of secondary importance. Of course, that is not what we see. People continue to be diagnosed with cancer, continue to even though mortality rates have decreased, this continues to be unfortunately for a number of individuals, a potentially life-threatening diagnosis. And because of the considerations that we were making before about nosocomial spreading of SARS-CoV-2, people have been very reluctant in seeking care, in going to the hospital. They've been very reluctant in maintaining their schedule for regularly planned preventative measures that provide early diagnosis for a variety of types of cancer. And so if there is one thing that it's very important to remind everybody, is that we've become very good at keeping the environment in hospitals clean, safe, and accessible to everyone, even against the backdrop of an ongoing COVID-19 emergency. So don't delay getting your colonoscopy every 10 years, don't delay getting your pap smear for cervical, as a preventative measure for cervical cancer, and especially don't delay your mammography, because we know that those things do save lives. And unfortunately, the National Cancer Institute has forecasted an increase in cancer-related deaths due to COVID-19, not because of the virus itself, but because people are not seeking, they're not maintaining their schedule for early diagnosis of potentially treatable diseases like colon cancer, like breast cancer, certainly cervical cancer.
So in your lab, you've been able to, and in the Wistar Cancer Center, we've been able to keep our scientists kind of spaced out at benches, and our cancer research, we're really lucky, has been able to continue during this pandemic under great leadership by your part. But I wanted to talk a little bit about some of the work that we're doing and we have been doing, especially as the themes you just spoke about have become so prevalent and so relevant. So cancer screening and having good early detection and diagnostics is just a huge issue, because the sooner we find it, the better we can treat it. And so one of the major missions of our NCI Cancer Center is early detection. And so I wonder if you could tell me a little bit about how we're doing in advancing cancer diagnostics, in particular around one area that I want to talk about now, and then I'll get into the other one, but our lung cancer diagnostics at Wistar. And how would the work that we're doing advance both the diagnosis and treatment for lung cancer?
Yeah, this is a very important, very timely area that we've been pursuing aggressively for a number of years. Obviously, lung cancer is a very common, very prevalent type of type of tumor, and therefore diagnosis. We know what risk factors are for developing lung cancer. Cigarette smoking is the most important one. There's no doubt about it. And therefore we know what individuals that are at high risk for being diagnosed with lung cancer, we know who they are. And therefore there's been a tremendous effort in developing, for instance, a spiral CT screening for individuals who don't yet have lung cancer but are at risk of developing one. The problem with spiral CT is that you see a lot of nodules in the lungs of these individuals. Only a small minority of those nodules are malignant. And so what do you do? I mean, how do you choose which one to biopsy and which one to leave alone, because they're just routine occurrences on a chest X-ray or a CT scan. And so the test that was developed at Wistar, it was designed specifically to answer that question. It was specifically designed to be combined with spiral CT, and in order to guide the surgeon and the medical oncologist potentially to biopsy nodules that might have a higher propensity of being malignant. This is a multi-gene platform that was developed here over the years, was validated extensively in multiple patient cohorts, hundreds of individuals, and continue to remain a very robust, easily administered test. It's a liquid, so the equivalent of a liquid biopsy. There is no tissue that is being collected. And might be useful, if validated in even larger studies, might be useful in really restricting the number of procedures that are being planned for individuals that are at high risk for developing lung cancer, which would be a tremendous step forward in the early diagnosis of the disease.
So you'd have earlier detection before you could see it. You would also have a quicker advance to actual treatment. So just overall, it would really be such a huge win if we can move all this forward. So there's another diagnostic that we're working on. Dr. David Spiker's lab is working on a biomarker, which is a kind of a blood test that tells you something about what's in your blood that would be used to detect ovarian cancer. So we have multiple diagnostics that we're pushing forward, but we're also working on therapeutics. And speaking of ovarian cancer, I was wondering if you could tell us a little bit about what Dr. Rugang Zhang's lab is, in particular to try and address standard of care when it's refractory for ovarian cancer and his research in that arena.
And that's also another very important, very timely area of investigation. Ovarian cancer is a difficult to treat tumor, and because it's very subtle. The symptoms are very non-specific. Unfortunately, women go to see their doctors when the disease has already progressed, because again there is really no early diagnosis. There is no equivalent of a pap smear or a mammography for ovarian cancer, not yet at least. And therefore, and some histologic variants of the disease are very dangerous. They are very life-threatening and unfortunately, as you pointed out, actually resistant to the therapeutics that we have. And standard of care in ovarian cancer hasn't really changed much in the last 30 years. There have been some advances, but relatively incremental, not the type of transformative advances, for instance, that immunotherapy has brought about in the treatment of patients with melanoma or even with lung cancer, where in fact immunotherapy now is actually first line of treatment as opposed to chemotherapy. So in ovarian cancer, we don't have such thing. We don't have newer approaches, and the work that Dr. Zhang has been pursuing is in fact designed to address specifically that gap. And what he has been interested in is in so-called epigenetics modifications in lung cancer. These are changes in the genome of the tumor cells that don't actually change the composition of the DNA, but change some modifications that take place on the DNA that affect profoundly the way genes are become expressed or repressed. And what he has found is that there might be what is called a synthetic lethality, meaning that tumor cells might become even more vulnerable by targeting, by inhibiting some of these epigenetic changes. And it's a very exciting, very new, potentially transforming development. It's specifically focused on the most aggressive form of ovarian cancer. And we look forward to actually clinical trials that will be carried out based on the discoveries that he made.
So there's a huge amount of work going forward in the Cancer Center, and as usual, I didn't get you to talk about your work because you don't like doing that, even though it's really interesting, and I think everyone would like to hear about it.
Right.
But we will now kind of shift gears a little bit to something that we've been very proud to have actually been able to advance during the COVID-19 pandemic, and I would say education's always been a critical part of who we are and what we do at Wistar. And in an era when division is rampant, this ability to unify and collaborate and partner is really critical for Wistar. We're small, but we're really good at reaching out to our networks to get things done and to build these bridges. And one of I'd like to start with you just kind of telling everybody a little bit about the long-standing education and training pipeline that's been established with our partners through life science training, and then a little bit about how it is developing a workforce pipeline in the burgeoning life science hub that is Philadelphia. So multi-pieces again, sorry about that.
Yeah, but it's very true. I mean, educating and training the next generation of scientists is part of our mission, is an important part of our mission. We think that we can do this with an approach that is really focused and at the same time encompassing that culture, the culture of discovery that is a trademark, if you will, is really a unifying identifier for Wistar. And so we think that we can bring to the table, to the education and training of our youngest colleagues, that mindset, that mentality of scientific method that we were talking about at the beginning of our conversation this afternoon. And so, obviously we are not a college, we are not a university, but as you pointed out, Anita, we also are very collaborative, and we believe that collaboration and partnership are really the ingredients to solve a very complex problems. And we see some of that actually, coming together in the SARS-CoV-2 emergency, when traditional rivals, particularly in drug companies, are actually working together, realizing that probably that's really the only way to make progress. We feel the same way, and when it comes to education. And so we had a long-standing partnership with the Community College of Philadelphia, 20 years.
Can you tell me a little bit, I'm sorry to interrupt, can you tell me a little bit about how that partnership started? Why did that partnership start?
Yeah, it was actually quite opportunistic, like many important things. Wistar back then had the problem of trying to retain quality laboratory technicians, people who would be well trained and happy to develop a career by working in a research lab, and that's a workforce. Obviously, it's absolutely essential for the performance and the success of research labs. And so, as part of trying to solve that need, we reached out to the community college, and we said, look, if you're interested, we could develop a path for your students. These are 2 years associate degree students. We would be happy to develop a path for your students, for them to come to Wistar, get exposed to science, to research, and have a mixed training that involves both a little bit traditional classroom work, if you will, but more importantly, hands-on laboratory training. And what started as an experiment turned out to be a great engine for the creation of a stable pipeline of research laboratories, not just for Wistar, but really for a number of other academic institutions, as well as biotech industry and large pharma. So that's really how that particular program started, and really continued to blossom, and became, to use your word, really as a pipeline for the development of new talents in the life sciences. And this is particularly important for our region, because if Philadelphia and the greater Philadelphia area wants really to become a hub for the life sciences, attract biotechnology startups, create a culture of entrepreneurship, at some point you need somebody in those labs. You need somebody that would staff those labs. And so creating a stable pipeline of a well-trained, well-educated workforce is an essential ingredient for the success of that idea.
And so then, once this program was established, you then took it a step further and established an apprenticeship program, which is the first national apprenticeship in biomedical research, and with help of both CCP and the Department of Labor of the Commonwealth. Can you tell us what this apprenticeship gives our students and why it's a little bit different than what everyone else is doing?
It's really a next step in the configuration of the original biomedical technician training program, which is the program we just talked about in collaboration with the community college. It really provides additional training, additional mentoring, additional hands-on training in terms of laboratory work and laboratory expertise. It helps the student develop an additional set of skills that would make him or her even more competitive in the workplace. And Anita, let me just say, the students that we're talking about are not the traditional, bachelor degree students. These are individuals who have families that are retooling their careers, that are starting a new careers, and individuals that are re-entering the workforce. So they need to have exposure to something useful that they can immediately put to work. And the apprenticeship represents really a runway of additional training and additional skill set development for these individuals to re-enter the workforce. And re-enter the workforce in jobs that provide a potential future career. They're well paid, they're stable, and more importantly, or just as importantly, they're local. They're here.
Yeah, I always have seen this as Wistar's best way to give back. It's what we do. It's within our wheelhouse, and it's so great that we can add to the community this way. And so now we're kind of building on this legacy and the biomedical training programs that we've established over the last 20 years, and we've created a new partnership with Cheyney University. I was hoping you could tell us a little bit about your hopes for bringing the first class of Cheyney students to train at Wistar in the spring, and what you hope both institutes are going to gain from this partnership.
Yeah, it's been truly exciting. I mean, the partnership between Wistar and Cheyney is very innovative. It's unique in our region. There's not been anything similar that was developed. And I think we're moving the goals a little bit with the Wistar-Cheyney partnership. We want to create, first of all, opportunities and tangible opportunities for students of color in the area of the life sciences and in the area of research laboratory training. And I think as an added goal of that partnership, and by the way, we're very excited that at our end, working collaboratively with the faculty at Cheyney, Brian and Christy managed to put together a very exciting curriculum that is ready to go. Depending, we're going to monitor what happens with COVID-19, but technically we're ready to go in the spring semester with the first class of Cheyney students to be trained at Wistar. But I was saying, we're expanding the goals for the Wistar-Cheyney partnership. This is not just about the life sciences, creation of a skilled workforce. It's really about injecting that culture of discovery as part of the opportunities for four-year bachelor degree students. We want my own personal goal, and I think we had shared objectives also with President Aaron Walton, the president of Cheyney, is really to make those students who are interested in STEM, in a STEM education, to making those students even more competitive than they are currently in tackling on graduate school, in tackling medical school, osteopathic medicine, in making them, as competitive or more competitive than their peers. And I think that this is a central objective that has to do both with the creation of a pipeline, but in my opinion, equally important, perhaps even more important, in the creation of the cultural shift, where STEM becomes, a lifelong objective for both education as well as career development. This is not just a pipeline for skill force development, but it's a pipeline for talent development. And I think that this is, in my opinion at least, is what I truly look forward to in the expansion of the Wistar Cheyney collaboration.
Yeah, I'm excited as well. Okay, 2 last questions. So number 1, what's keeping you up at night? And you can't say everything, that's not an answer.
No, that's not an answer. I honestly think a lot about COVID-19. I really do. I think a lot about, what's next. Now we're coming down to the wire, and a number of those, as I mentioned before, a number of those phase 3 trials will be coming to a head. I remain, and I don't want to talk politics, of course, but this is a difficult problem to solve. We have seen a number of therapeutics that had hopes for COVID-19 patients turn out to be ineffective in the clinic. The antibody cocktail that was developed by Eli Lilly unfortunately showed no benefit in the phase 3 registration trials. We hope that the Regeneron cocktail of neutralizing antibodies will fare better. We talked already about hydroxychloroquine and convalescent plasma. And unfortunately, even remdesivir, a drug that was developed for Ebola, for the treatment of Ebola, is a nucleoside analog. There was a lot of anticipation about remdesivir, and as in a global phase 3 trial, global means global. It was run in 500 sites in 30 countries, 12,000 patients. I don't know if there is a trial that was bigger than that in immunology and virology. It didn't really show much benefit. And remdesivir has now been approved by the FDA for use in the United States as standard of care. And I don't understand it. I don't know if anybody does, but I certainly don't. And so those are a lot of things that keep me up at night thinking about that.
Yeah, so behavioral health, everybody. Wear your mask, keep your distance, don't gather in groups, behave.
That's the only thing that we have right now, and it's the only thing that we know it works.
Right? And then here's my last one. What are you optimistic about?
I think that we will have a successful and safe vaccine, probably more than one. I'm really confident on that. I think that this will not be the end of the COVID-19 emergency. I think that there are still a lot of things that we don't know about this virus. This doesn't behave like a respiratory virus. There are things that happen to severely ill COVID-19 patients that have nothing to do with the respiratory virus. There is a strong inflammatory component, there is a strong vascular component that we don't see with other viruses that attack the upper and lower respiratory tract, for instance, the flu. So I don't think that having a vaccine that is safe, most important thing, right? Because you give a vaccine to healthy individuals. I don't think that having a vaccine that is safe and effective will solve the COVID-19 emergency. I think that COVID-19 is here to stay. But I am hopeful that will generate additional knowledge, that we will learn more from it and being able to control it better in the upcoming months.
Yeah, I totally agree. So with that, I just want to say thank you so much for sharing your insights. I think it's been really interesting. We're kind of this little place on the corner, and not everyone knows what happens on the inside. So giving everyone a sneak peek is, I think, a great thing to do, and I appreciate your time. And if you have any, we've run out of time, but if you have any questions, please just email them to me, apepper@wistar.org, and I'm happy to follow up with any of you. And otherwise, Dario, have a wonderful evening, and I wish you all a wonderful evening, and please be safe.
Thank you, Anita. Thanks everybody for joining us tonight.
Thank you.